Ready to rock, are you ready to roll? Will you open the lock to your heart and your soul? Are you feeling the beat? Is it turning you on?
Here we go. This is Biologically Important Molecules tutorial. There is a worksheet on my website, Zerobio. I'll put a link in the information section so you can get the 5-page PDF for this. As I go ahead and go through it, you can fill it in as well. Now, one thing you're going to notice is that I'm using a colored series of images, and yours will be not colored, and you're welcome to color it in the same fashion that I've got, and I'll talk to you about that in a second. All right, so go ahead and get the worksheet, and then come on back here for the video. All right, so as you can see here, I've got some colors in my diagrams. We're looking at carbohydrates, which is one of the 4 major types of biologically important molecules. There are carbohydrates, lipids, proteins, and nucleic acids. I'm not going to be doing nucleic acids in this video tutorial, however. All right, so as you can see here, I've got some molecules here. Carbon is typically black, and I'm getting that color from the molecular model kit colors. So if you want to go ahead and color yours in, you can do that. And of course, you could choose different colors if you prefer. But carbon here is black, oxygen is red, hydrogen is 1. And as you've probably seen in my functional group tutorials, hopefully you've seen some of those, carbon makes 4 bonds, hydrogen makes 1, and oxygen makes 2. And of course, we'll deal with nitrogen when we get to the amino acids in a moment. Now, this is a big section in the in the biochemistry strand, biologically important molecules. Typically, you would probably hit this a few weeks into the first strand on the biochemistry strand. And, you know, it's a lot to learn. It's a lot to memorize. There are a lot of structures, and different teachers will have you either memorize structures or, you know, just get familiar with them. You know, the more I think that you can memorize, the better off you're going to be. All right. Also, of course, you need to understand them. So what I'm going to do is I'm going to be revealing some information that you can write down in these spaces. So the first thing is A and B. Now, what we're getting at here are the types of reactions that either produce the larger molecule here on the right-hand side, so that would be this arrow with this label A, condensation, as a condensation reaction, or B, taking this larger molecule and breaking it down into these smaller pieces here. So what we have is a monosaccharide here. This is also a monosaccharide. Both of these are glucose molecules. So I'm going to reveal this. We're going to write glucose in there. So this is a glucose molecule, and this is a glucose molecule. These are in the category of biologically important molecules called carbohydrates, meaning they are composed of carbon, hydrogen, and oxygen. They're not hydrocarbons, they're carbohydrates. So there's oxygen in there as well. And you can see that glucose has a ring structure here, which has 6 atoms. 5 of those atoms are carbons, and 1 of those atoms is an oxygen. The formula for glucose is C6H12O6, right? So there are 6 carbons in here, there are 12 hydrogens, and there are 6 oxygens. There is a particular way that all of these atoms are oriented. So for example, you can see this is a hydroxyl group, and this is why we studied that in the functional group stuff. There's a hydroxyl group here, there's another hydroxyl group here, there's a hydroxyl group, this is a hydroxyl group here, and there's another hydroxyl group here. All right? So, you know, we're doing biologically important molecules, but we're already identifying, you know, functional groups in it. Now, there is another functional group in here, and I don't know if you can see this or not. Check this out, COC. Within the glucose molecule itself is an ether. Okay, now that's going to be important because when we form a bond between 2 glucoses, we're going to produce another ether as well. Okay, but that's not the same ether here. So these are the functional groups in glucose, and in particular, the most important part is, and I want to switch to another color, is going to be this hydroxyl group in that glucose. Now, of course, there's a hydroxyl, 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 and another ether. But when 2 of these glucose monosaccharides, single sugars, are close together, the hydroxyl groups, this hydroxyl group on this carbon and this hydroxyl group on this carbon, they react. Now, you might remember I said in a previous functional groups tutorial, it's the functional groups that determine sort of the reactivity of the molecule. And here we're going to see that. This is a reaction between two hydroxyl groups, and it's called a condensation. In other words, we are going to condense together two monosaccharides to produce a bigger molecule, which will be on the right here, which, let's reveal the name for that right now. This is a disaccharide. Two monosaccharides linked together make a disaccharide. Now, this disaccharide that will be produced from the condensation of two glucoses is called maltose. And you probably learned about that in grade 11 biology. That's why I've chosen this example. So, glucose plus glucose produces, through a condensation reaction, maltose. But another molecule will also be produced and is over here on the product side, and that's this one, which is water. So, I'm going to get rid of that stuff, and I think I'll stick with the blue, and let's have a look at this. In order to produce water, water cannot be just produced out of nothing. The atoms of the water have to come from the molecules that are reacting. And here we can see, if we were to take this hydroxyl group right there, and this hydrogen, and it probably would be a decent idea for you guys to circle that stuff. If we were to pull that off, imagine the molecules are bumping into each other. As they collide, the water falls off or breaks off. That would form H2O. That stuff would be gone. What's left is this oxygen right here from this glucose molecule. This oxygen would then be bound to this carbon because this is going away. Don't cross that stuff off. This is going away. That's going away. All of that is the water. This then, as the two molecules collide, would be attached. Now, that is going to require an enzyme that will perform this reaction, this condensation reaction, linking them together. Now, can you see on this side what the linkage would be like? It would be carbon to oxygen to another carbon, just like this, carbon, oxygen, carbon. Carbon, oxygen, carbon. Or let's go back over here and let's circle this right here. Well, here. This was the carbon from the glucose on the right, so it would be this carbon and its oxygen and another carbon, and that is an ether between the two molecules when they came together. That's an ether, and so this is called an ether linkage. I mean, it is a functional group within maltose, the disaccharide maltose. It is an ether functional group. But we know that it was formed from the union of two monosaccharides, so it can be called an ether linkage. Now, if all you have is maltose and you look at it, you may not know, like, well, how do I know, that it was formed a minute ago and so it's a linkage? It doesn't matter. It's an ether, all right? But maltose is made up of two glucoses, and so what links the glucoses together in the maltose molecule is an ether linkage, or just an ether, all right? So in the condensation reaction of these two specific monosaccharides producing this particular disaccharide maltose, an ether linkage is formed, and water is also produced. So this reaction is also called a dehydration synthesis. We are synthesizing maltose, and we're doing a dehydration. We're removing water from this side. We're producing water. So it's a dehydration synthesis or a condensation reaction. All right? So already in biologically important molecules, we are distinguishing between monosaccharides and disaccharides. Now, if you were to eat this molecule, like when you've eaten starch, and amylase breaks down the starch into maltose, maltose then, gets broken down into glucose by the enzyme maltase. Well, now we're looking at reaction B, the hydrolysis. A hydrolysis reaction is a reaction in which the disaccharide is split and two monosaccharides are released. The two monosaccharides would be these glucoses. And water is a reactant in this reaction in a hydrolysis. Hydrolysis, if you look at the name, really means hydrolysis. Lysis means splitting. Hydro refers to water, so splitting with water. And so what really happens is one of the atoms, hydrogen, will go on to the oxygen to form this OH group. So just imagine this hydrogen clicking onto the oxygen, to form a hydroxyl group. This OH will go on to the carbon, forming the other hydroxyl group. And they are now separate from each other. All right? So we can split disaccharides by hydrolysis reactions, which are essentially the reverse of condensation reactions. All right? And the linkage that we are splitting with water is an ether linkage. If you take an ether linkage and put water into it, you generate two hydroxyl groups, and of course, the rest of the molecules. So, there's a little bit of review on monosaccharides, disaccharides, condensation, and hydrolysis. The other category of carbohydrates is polysaccharides. Poly meaning many. So this is many saccharides, many sugars linked together. An example of a polysaccharide is down here, glycogen. Now, for these, I have just drawn a little bit schematic diagram for the ring. It is the same as this molecule, this glucose, but I've drawn it like this. The red dot would represent the oxygen in the ring. And this spot here looks like just one atom. I'm really sort of saying that this is where the atoms are joined together. I mean, you should know if this is glucose and the next one is glucose. If all of these are glucoses, you should know that that is going to be an ether linkage. That's an ether linkage, another ether linkage, and that would be a hydroxyl group ready for another one. All right, but this is a schematic diagram. We're trying to keep it simple. The thing to note about glycogen is when the body has enough glucose, all right, coming into the body, you use glucose as your main energy source. You will study very soon a process called glycolysis. Here, let me write that word. You don't have to write this on the sheet, but you're welcome to if you want to. Glycolysis. Now, that may be a new term for you right now, but soon, and I've got video tutorials on this, this is going to be very familiar to you. Glyco for sugar, lysis for splitting, just like hydrolysis. This will be the splitting of sugar. What I'm saying is your body uses glucose, puts it through the process called glycolysis, and out of that comes energy in the form of ATP. Okay, something to be studied later. But if you have enough glucose in the body and the body says, I don't need to make any more ATP right now, thank you, store this up for later, all right? Your body goes, well, how am I going to store glucose? It's all these individual monosaccharides. It's more convenient to package them all together in a big molecule called glycogen, but where they're squished in together, and that gets stored in muscle and in liver. And that is the purpose of glycogen as a polysaccharide. Many sugars click together. And when you're ready, the body will just say, I need more energy. So, you know that stored up glucose you have as glycogen? I need you to split the glycogen, do hydrolysis reactions at the ether linkage to release the glucoses so that I can use them for glycolysis. So that's how that works. So glycogen is an example of a polysaccharide in animals. Now, another polysaccharide, and by the way, these are both polysaccharides, so write polysaccharide. Another polysaccharide is found in plants, and this one is cellulose. Now, starch is also a polysaccharide. It's very similar to glycogen, and possibly in the course you'll learn some of the differences. Cellulose is another plant-based polysaccharide. It is a storage form of glucose. And remember, plants produce glucose through photosynthesis, and plants also do glycolysis. They also need energy to grow and to reproduce. But when the plant says, I've got enough energy, I'm going to store this stuff up. In fact, glucose is polymerized, clicked together in chains of cellulose, this molecule, to produce structural materials. Cellulose is in the cell wall of plant cells. Remember sort of the rectangular shape of plant cells, and you may have learned that cellulose is in the plant cell wall. So plants make glucose by photosynthesis, and then they put it into their cell walls in the form of this polysaccharide, cellulose. Now, cellulose is very similar to glycogen, except each glucose is flipped differently. So this one is right side up. This one is upside down. Right side up, upside down. See the way I drew the oxygen there and the little flag-like structure there? So that's a key difference between animal polysaccharides and this particular plant polysaccharide, cellulose. And we don't have the enzyme cellulase that can break this bond. Because of the orientation of those glucoses, we actually can't break it down chemically through enzymes. So this is why cellulose in plants like lettuce and other fruits and vegetables tends to act as roughage in our digestive tract. We don't get the glucose energy from it, but it will sort of scrape the walls of the intestine. It helps clean out that stuff. So, monosaccharides, disaccharides, polysaccharides, there are many more of these monosaccharides, disaccharides, and polysaccharides. I just chose representative examples. That is our first category of biologically important molecules called carbohydrates. Now, let's go to the next page. Have a look at that for a moment. I'm going to look at proteins next because they're a little bit simpler in terms of their individual structures than lipids, but we'll do that in a moment. Proteins are big molecules. Many enzymes are proteins. So they can be quite large molecules, but the individual bricks, the building blocks, just as in carbohydrates, the building blocks were monosaccharides, which are clicked together to make disaccharides, which eventually are polymerized into long chain-like molecules, which are polysaccharides. Proteins are like polysaccharides. They are polymers. The individual bricks, the monomers, monomer, single building block versus polymer, which is the chain. This would be an example of a polymer. A polysaccharide, glycogen, is a polymer. The individual subunits, mono, are monomers. So the monomers for proteins are amino acids. This is a representative amino acid, and so is this. Now, in these models, I have drawn carbon in black, oxygen in red, note the double bond, hydrogen in white, nitrogen in blue, nitrogen makes 3 bonds, all right? And this R represents a variable group. It's not one atom, it's a group of atoms that can vary depending on which amino acid it is. So this would be one type of amino acid, and although this molecule looks the same, it could be the same, or it could be a different amino acid depending on what atoms were in this group. But they can be quite large, and it would be a drag to draw them. So this is a way that we sort of shorten it. The other thing to note is that amino acids, inherent in their name, have 2 functional groups. And you're welcome to circle this. This would probably be a good idea. That is the amino group, and we studied that in functional groups before. And this is a carboxyl group or acid group, so amino acid. And this one has an amino group here and the acid. So amino acids are the building blocks of proteins. And in order to make proteins, you have to click the amino acids together. They have to react. You can see that I've drawn the carboxyl group here in close proximity to the amino group of the other amino acid. It's always carboxyl reacting with amino. It's not carboxyl with carboxyl or amino with amino. Carboxyl and amino. The type of reaction that's going to occur over here is the same as we saw for the carbohydrates. A condensation reaction will occur to produce a molecule over here and water. So it's condensation or dehydration synthesis. As well, this molecule can go through hydrolysis, and I'll show you where in a moment, to produce the amino acids again. So you can break this bigger molecule down to its individual building blocks or monomers. So what is produced when two amino acids come together? It's called a dipeptide. There are two amino acids here, although water has been removed from them, so they're missing a few atoms. All right? This is a dipeptide. If another amino acid was clicked on the end, it would be a tripeptide, and etc., etc. As you build up a chain of amino acids, at some point, it's going to be called a polypeptide, like polysaccharide. All right? So amino acid and amino acid, dipeptide, tripeptide, eventually polypeptide. And if that chain of amino acids, like I've shown over here, all right, I've illustrated AA1 for amino acid 1, clicked onto amino acid 2, 3, and some of them can repeat. They don't have to all be different. They can be some similar ones. Eventually, if this is sufficient number of amino acids to be functional as a molecule, it's called a protein. So a polypeptide or a peptide can have amino acid and amino acid and amino acid and amino acid with lots of different crazy shapes like that. Let's say they're all joined together. If this is enough to do the job, in other words, it's a functional molecule on its own, it's called a protein. If this chain of amino acids, called a polypeptide, needs additional modification, or another polypeptide there and maybe another one there, together all those polypeptides, then the whole thing now is called a protein. Now that will become clearer to you, I think, a little bit later on, if that's a little bit confusing. So let's talk about how these amino acids react, these biologically important molecules. Well, we need to produce water, and we're going to get water from these groups. So the hydroxyl group of 1, and this is a good thing to circle, the hydroxyl group of this carboxyl, maybe it's confusing how I'm saying that, but this is a carboxyl group, and the OH, the hydroxyl part of that, and the hydrogen will come together, and that will produce our H2O, that will be water, will come off. So imagine now that, don't write this, that oxygen's gone, that hydrogen's gone, that hydrogen's gone, that made the water. You now have C double bond O left, hanging sort of free, and NH hanging free. Over here in this molecule, see the way there we have the C double bond O and the NH? Now, I've drawn them straight up instead of curved. So, I mean, imagine this. Imagine there's the carboxyl on one side, there's the amino group on the other side. When they collide and water comes off, can you imagine that they kind of bang into each other and go straight up like that? It's not a critical thing to know, but just that's a way that you could look at it. So, we have the remnant of the carboxyl group here, the remnant of the amino group here, and this is where the molecule has been linked. Now, this is not an ether linkage anymore. Now, I'm wondering if you can see this. I need to draw something up here. I'm just going to do R for a variable group. If you were to look at this molecule in the functional groups tutorials, we would have highlighted this and called this an amide, right? That's the amino part, but joined to C double bond O. This is an amide, and that's why you learn those functional groups. So it builds to the next topic, which is this one, all right? I'm going to erase all of this stuff. So this area right here is very similar to the structure I just drew. The R part could be this, all right? And, well, what we're missing is another H, right? And then you have the amide. Well, when they come together and react, that H disappears. And now we have this. And so either the whole section or this part right here is called an amide linkage. So it's not an ether linkage anymore, it's an amide linkage, all right? But it's more commonly referred to as a peptide bond, A peptide bond is a bond between the carboxyl of one amino acid and the amino group of another. When they come together, this is called a peptide bond. So, in this protein down here, the bond between amino acids is a peptide bond. All of these would be peptide bonds, carboxyl reacting with amino group. Now, if you wanted to split this molecule, the water would go in at the peptide bond. Imagine this OH clicking onto here, OH, you don't have to draw that if you don't want, and that hydrogen going on to there, that would regenerate the amino group and the carboxyl group. So that would be a hydrolysis, and you now have your amino acids. Now, why would you need that? Well, when you eat a piece of chicken, all right, or hamburger, and you get protein, or whether it's plant-based proteins, peanut butter, beans, whatever, your body will take those proteins and split them into individual amino acids, and then you will then assemble those amino acids into the proteins of your choice to make your muscle. I mean, because if you eat chicken muscle, right, you're eating a piece of chicken, that's the muscle you're eating, right? As you eat that chicken, here, let me draw it here. There we go, big piece of chicken there, chicken leg. You're eating that Kentucky Fried Chicken, Swiss Chalet, whatever it is, right? You eat that. You're eating protein. Yeah, you're getting fat and carbohydrates and all that. You're eating protein. You don't necessarily take that chicken protein and just stick it into your arm and get big muscles. Instead, you break down the proteins of the chicken and take the amino acids, and your body says, okay, now human muscle protein has to have amino acids in this order as opposed to chicken. You are not a chicken, right? You are a human, right? So you will arrange those amino acids in your order and make your protein. So this is why you need to eat protein. You have to build your own proteins, but you split them by hydrolysis into individual amino acids, and your body puts them in the correct order to make human protein. Okay, so proteins are built from amino acid monomers to make the protein polymer. Proteins are enzymes in the body. Many hormones that you'll talk about later are made of proteins. Various structures of the body are made of protein. Protein can also be used for energy, but not very often. So we have carbohydrates and we have proteins, two of the four major types of biologically important molecules. Now, feast your eyes on this one. This is some of the category of lipids or fats. In the lipid category of biologically important molecules, there are fats, oils, waxes. There is steroids, steroid hormones, cholesterol, phospholipids, parts of the cell membrane, and I'm only going to talk about a couple of those, and that will be the lipid category. Now, have a look at the molecule that we've got here for you. This is 3 carbons. I've had to stretch them out so that there's enough room to show what we need to show, but that's fine. This is still a single bond. It's a big stretch, single bond. OH, OH, OH. This is a type of alcohol, and specifically, this is glycerol. Now, notice the ending OL, alcohol, glycerol, propanol, ethanol, all right? Alcohols end in OL. This molecule has 3 hydroxyls. Now, you don't have to circle that yet, all right? 3 hydroxyls. The rest are carbons and hydrogens, 4 bonds around carbon all the time. All right, so to make the lipid I want to talk about right now, we're going to make something called a triglyceride. Tri for 3, triglyceride, 3 for something, that's the tri, and glyceride referring to the glycerol molecule that it comes from. So it's going to be a glycerol and 3 other things coming together, all right? And that's what I'm showing you in the diagram. Glycerol, and here are 3 molecules. There is a subtle difference between them, all right? 3 molecules that will react with that glycerol. So I'm going to erase all of that and show you the name of this. These are called fatty acids. Now, They're referred to as acids because they all have the carboxyl group, which is the acid group. All right, so carboxyl groups make the acid part. Fatty refers to this carbon-hydrogen chain. Now, I've drawn a very short chain. Usually, there's something like 16 or 20 or 30 carbons. Very long, complicated. When there is something made of carbon and hydrogen, so much of it, that stuff is very non-polar and hydrophobic. Now, you may or may not know that word, hydrophobic, fear of water. It doesn't dissolve in water. It's non-polar. So it's, fear of water, doesn't like water. It's hydrophobic. That stuff is referred to as fatty stuff. Now, I'm not necessarily talking about the fat on your body, although some of this is in there. Just take my word for it for the time being. So you get the carboxyl group and you get the fatty part. Now, it's not critical that you understand that completely or that you learn about that this is a fatty part, but it is. And there's the carboxyl, which I think is relatively easy to understand. Now, note that all of this has carbon to carbon and carbon to hydrogen single bonds. All of the spots on the carbon are filled with hydrogen. Carbon makes 4 bonds. It cannot accommodate any more hydrogen. Look, there's 1, 2, 3, 4, 5, 6, 7 in this molecule. Come down to this bottom one here. Notice there's only 1, 2, 3, 4, 5 hydrogens. This one has 7. This one has 5. This one could accommodate more hydrogen if it didn't have a double bond there. In this case, there's a double bond between these two carbons, so it can't form another bond with hydrogen here or there. Carbon still makes 4 bonds. This is called unsaturated. This is an unsaturated fatty acid, meaning it's not fully saturated with hydrogen. These are saturated fatty acids. They are saturated. They are holding the maximum amount of hydrogen they can. So unsaturated fats or fatty acids have double bonds, at least one, all right? Double bonds between carbon and carbon. We're not talking about this double bond. It's between carbon and carbon. They're called unsaturated fatty acids. They don't have all the hydrogen. Now, we'll talk about why that's important later if I don't forget. Anyhow, I've got 3 fatty acids here and 1 glycerol molecule. Now, all of these will perform condensation reactions or hydrolysis reactions in reverse. So, in order to do the condensation reaction to produce the product we'll talk about in just a moment, we're going to have a reaction between the functional groups, between the carboxyl group here and the hydroxyl group here. Now, for our purposes, it doesn't really matter which exact atoms react, but maybe to make it a little bit easier, you may already know that acids release hydrogen ions in solution, all right, like HCl, produces H plus and Cl minus. It breaks off and gives the H plus, okay? So let's say this. The carboxyl group will release its hydrogen here, and the glycerol will release its hydroxyl group there. I mean, I could have taken the hydrogen from here alone and the OH from there, but I'm going to take the H off. It's not really critical. And H and OH interact, imagine as they collide together. And this one, H and OH, H and OH. And that makes 3 waters. And that's what we see over here on this side. The 3 waters as products, right? This is a condensation. This is a dehydration synthesis. As this carboxyl and that hydroxyl collide and react, water comes out. And what happens? Imagine this is gone, and this is gone, and this is gone. This O will then attach to this carbon, and that's what we see here. Now, I've drawn the molecule a little bit different in order to accommodate these molecules. There's the fatty acid, there's the carboxyl group, and that O is attached to the carbon now. This was the O from the carboxyl, and this carbon from the glycerol. The same thing happens here. Those go away, this oxygen clicks onto the C. Those go away, this oxygen clicks onto the C. In every one of these cases, this molecule that is produced out of 3 fatty acids and 1 glycerol is called a triglyceride. 3 fatty acids on 1 glycerol backbone, it's sometimes called the glycerol backbone, the glycerol molecule. These are lipids. This is a lipid, all right? And now, the linkage that is produced from a carboxyl and a hydroxyl interacting is this. C double bond O, carbon on one side, oxygen carbon on the other. That right there. Ester. This is a ester linkage. Now, if you just take the triglyceride itself, you say, well, there's an ester functional group. But it happened to be formed from glycerol and fatty acids coming together, so it's also an ester linkage. It doesn't matter whether you call it ester linkage or an ester. All right? So in all of these spots here, we have another ester or ester linkage and another one. Okay? Now I want to show you one quick thing, and this is going to just confuse the heck out of you. Some people just call this the ester. All right? Just the C double bond O and the O. All right? And whatever, 6 of one, half a dozen of the other. At any rate, we now have a triglyceride with ester linkages, 3 of them, 3 waters, and this triglyceride is another way to store the building blocks of fats. You know, let's say in your diet, you get, glycerol, or you get some fatty acids in your diet. You can click them together to make this big globular molecule. And a fat, imagine a chunk of fat on a steak, all right? I'm not going to draw a steak here. There's your steak, Each chunk of fat in the steak is going to be thousands or hundreds of thousands of these triglyceride molecules in a big blob, all right? And when you eat that, you chew it, physical digestion, when it goes down to your stomach, gastric lipase and into the duodenum, pancreatic lipase, remember that enzyme from grade 11? Lipase will split this molecule, perform a hydrolysis reaction. They're all doing hydrolysis. Lipase acts on lipids. Triglyceride is a lipid. And it will split these molecules right there. All right, so if you take this H from water, click it on there, and take the OH and click it onto here, you will regenerate your glycerol. And you will have fatty acids if you do that at every spot. So when you eat fats, your body will take some of that fat and store it, because this is a great storage form of energy, because what's going to happen is when you need energy, let's say you're running a little bit short of glycogen and you don't want to use your proteins right now because that builds up muscle and body parts. Fat, we've got fats. Like, why do I have this fat? I can burn some of that for energy. So what you do is you take it. Lipase will split the fat into glycerol, and for every triglyceride molecule in here, remember we said thousands, hundreds of thousands, for every one you get 3 fatty acids, all right? These molecules will also go into cellular respiration, like glucose goes into glycolysis, all right? They will go in and they will create lots of energy, or ATP, and you'll study that in the metabolism unit. So fat is a very good source of energy, all right? And that's because you get 3 fatty acids in every fat molecule, every triglyceride. Okay, more on that stuff a little bit later. But my point here is, this is why we're doing the course in this way. Start with atoms, molecules, chemical bonds, basic, simple stuff, right? Then you get into that polar stuff. It's like, oh, this is getting a little tricky. And then functional groups goes, oh my God, I got to learn all these functional groups. And then into biologically important molecules. And your head's starting to spin. You're going, is this biology class? But we need all of this to build into that next strand, which is metabolism. And wait till you see the biochemistry there, all right? It's pretty complicated, but it's very cool stuff, and it all depends on getting a good foundation in this stuff, okay? So that's condensation and dehydration synthesis and hydrolysis for lipids. Now, this is just one type of lipid or fat. This is called the triglyceride. I want to talk about one other type, and it's called the phospholipid, all right? So again, we have the name lipid, but now we have phospho, which will imply phosphate or phosphorus. And here we can see P over here. All right, now, what we have here is a triglyceride. people often go TG as the short form for triglyceride. Remember, it has the glycerol backbone, fatty acid here. I happen to choose the one that was unsaturated, and the saturated fatty acids. It doesn't matter which ones particularly. And we are going to convert this triglyceride into a phospholipid. Now, phospholipids are part of your cell membrane, all right? And you're made of billions or trillions of cells, each one of them with cell membranes. And by the way, not just the cell membrane, the outer membrane, nuclear membrane, mitochondrial membranes. There's two of them, outer and inner. Chloroplast membranes, the membranes of the endoplasmic reticulum. All these membranes, the building block that makes up the membrane, the brick, is a phospholipid molecule. So if you were to look at, here's a cell, okay, and there's the nucleus, okay. If you were to take a small piece of this, all of the individual molecules that make up this membrane or wall are these things. All right, now I know you can't probably picture how these things fit together, but just imagine this molecule. I don't know, imagine it's a big square like that. Just imagine a bunch of them joined together. All right, it's not exactly how it is. Joined together to make the membrane. So phospholipids are important in the body, and you create them from the fat in your diet. So don't let anybody tell you that, oh, I don't need to eat fat, or fat's not good. You need fat. All right, you need fat in your diet, just not too much. And you will use some of the fat in your diet for energy, as I said previously, and you use some of it to make these molecules called phospholipids. And now, let's do some labeling here. Oops. This molecule down here is called a phosphate group. So let me just do that, phosphate. So go ahead and label that. It's got a phosphorus atom, and it's got its 3 hydroxyl groups, and you will have seen this before in the functional groups. Normally, you will see a double bond O on a phosphate group. This particular phosphate is modified. There's an extra carbon group on there. It's a big thing that I didn't want to draw out, and you're probably grateful that I didn't. It's called choline. So it's a little bit more complex than this, but we don't need to know that because the name is phospholipids. This is where the phospho comes from. So how do we go from a triglyceride to the product, which is phospholipid? And by the way, I'm not including any waters that might have to be used or released in this reaction. It is actually a bit more complicated. So this is our phospholipid, and as you can see, the phosphate group is attached right up here where this fatty acid was, and the fatty acid is over there. All right, so here's what essentially happens. I'm going to move something. You can't do this on your diagram, but an enzyme comes along and breaks this fatty acid off. Okay, this was attached. It breaks it off, and I'm just going to put this one over here, and I'm going to get rid of this one because it will be the same thing. So just tap that. Okay, so there's the fatty acid that was here. The enzyme breaks off one of the fatty acids, and it takes this phosphate group and it clicks it on. Now you'll notice that the hydrogen comes away. All right, that's on purpose. The phosphate goes there. See, it looks just like this, right? So that's what happens. Remove a fatty acid, so you have just glycerol like that, and 2 fatty acids, and in the 3rd spot, click on a phosphate group, and this hydrogen can go over here to regenerate the fatty acid. We now have a phospholipid molecule, okay? So a triglyceride, remove a fatty acid, click a phosphate group on it with a little bit extra stuff we're not talking about. This is called a phospholipid. And, you know, you get a fatty acid out of it. This is the building block, the main building block of the cell membrane. And this belongs in the category of lipids. Beyond this stuff, there are steroid molecules. All right, there are the sex hormones built from steroid molecules and cholesterol, and etc. Now, I'm going to talk a little bit about this stuff feeding into energy pathways. So we'll go to the last diagram. This is a very basic diagram of some of the stuff to come in metabolism. All right, this represents the process called metabolism, which are the chemical reactions that break down molecules, essentially food molecules, to produce energy. The energy is in the form of ATP, adenosine triphosphate. And here I've illustrated A for adenosine and 3 phosphate groups. We don't add the other atoms, we just draw the P's. This is a high energy molecule like a battery. You'll learn about its function later. But the whole purpose of this metabolism is to produce this energy molecule. Thousands and thousands and thousands of them every second of every day. All right? What molecules go through metabolism to make this energy? Mostly carbohydrates. All right? And in particular, glucose. So if you eat chocolate cake and you get sugar in there, you're going to get polysaccharides. You're going to do hydrolysis reactions and split them up eventually to get monosaccharides, glucose. That will go down this process called metabolism. All of the other carbohydrates that you may have heard about can feed into this at some position. As well, lipids. Lipids can be used to generate ATP as well. And you'll learn that in the metabolism strand. Okay, so of these biologically important molecules, carbohydrates and lipids can be used to produce energy. Now, they may also be used to form structural components or parts of the cell membrane or whatever, or storage forms, but they can also go through metabolism. And of course, proteins can as well, although they play a pretty minor role in energy production. All right, but that's the idea. Now, those are 3 of the 4 categories of biologically important molecules. And that's not all there is in carbohydrates, lipids, or proteins. This is just a little bit of biochemistry to help review some of the fundamental stuff. The 4th type of biologically important molecule is the nucleic acid, DNA, RNA, and normally that is studied in the genetic strand. You might do a little bit in this strand as well, but I'm not going to cover that right here. But that gets into the nucleotides, deoxyribose, and the nitrogenous bases, and the phosphate again, and the structure of DNA. Nucleic acids are not used to produce energy, all right? So when you eat that chicken, you are getting protein, which you can use for energy, or you can build your own proteins, enzymes and hormones and other body parts. You're getting carbohydrates. You can store that for later. You can make up parts of your cell. You can use it as the main source of energy. And you get fat. Of course, there's fat in chicken or steak or whatever. And you're going to store that. Fat is sometimes used to insulate body organs. You can also generate energy. But in the cells of that chicken, you're also going to get the nucleus. As you eat the meat, you're going to break the cells open. There's the nucleus, the genetic material of that chicken. That genetic material is not just going to integrate into you when you're part chicken person, right? You will then take the DNA, let's talk about DNA, of the chicken, break down all the DNA into its building blocks, into the monomers, the nucleotides, adenine, thymine, cytosine, guanine, those nucleotides, and you will then build your own human DNA in the order you want. So your body will say, I need an adenine, and then another adenine, then a thymine, and then a guanine, and another guanine, and then a cytosine, and, in your human order. But you will take the chicken DNA and make your DNA. You don't take a chicken gene or stretch of DNA and use it in you so you're part chicken. You are part chicken only in that your DNA is actually made from the stuff that you've eaten. Like, where else do you think your DNA comes from, right? You start as a single cell, as a zygote. Sperm comes with some DNA, 23 chromosomes from dad, 23 from mom. You got 46 chromosomes, but that's one cell. And then you split into another. You needed twice as much DNA, right? Think of mitosis, right? The DNA replicates in late in interphase. 46 become 92 chromosomes, each going to the side, 46 each. All of a sudden you have twice as much DNA. Where did it come from, right? It came from the food, from what you took in. And so, you had salad, you had meat, you had bread, you had cheese and milk and all that. And all that stuff's got DNA, and then you break it down to nucleotides, rearrange it into a human order, so you are a human being and with human genes. All right. Whereas the other stuff that I've talked about, carbohydrates, lipids, proteins, is used for structural materials and also energy production. All right. So I know that was a long tutorial, almost 50 minutes, but hope that helped you out. Take care.
So are you ready to rock? Are you ready to roll? Will you open the lock to your heart and your soul? Are you feeling the beat? Is it turning you on? Can you carry the heat now that summer is gone? I can see in your eyes, a ghost never dies. It's abundantly clear. You're gonna rock the school year.